All right, so if people have this up and nice, we can start looking at some of the things we talked about during the motion registration. So if people still have it running, that's fine. We can all just watch together. So if you said keep movie raw, normally you probably don't want to do this because you don't need your raw data. You have it in the TIFFs, it's fine. But if you want to keep the raw movie, you can then watch the raw movie on this left side with the registered movie on the right. And so this is what this looks like. Oh, yeah, sorry. I should be more clear, huh? Okay, so if you press registration, there's two things. There's the view registered binary, and then there's also the registration metrics we talked about earlier. And then, oh, great, okay. Then here, this is this window here. And now we can play the movie, and we can see.
If you didn't finish processing, you can just load from Carson's folder the results.
Oh, that's, you think they have access?
Everyone has access.
Oh, okay. All right. So do you see this thing here? So I'll show you how to load it. Actually, I'll just do it again because it won't take too long. All right. So if you want to load it, you would take File and then say Load Process Data, and now you have to go to Groups Hackathon Data. I'm guest 23. Example data Suite 2P, and then what Suite 2P looks for. So there'll be multiple planes in this folder. In this case, we only had a single plane recording, so there's just one folder. And then what Suite 2P looks for in the GUI to load it is the stat file. So the stat file has like all the cells and information where they are and that sort of thing. That's going to make all the masks for it. And it's also going to use all the other files in that folder. So basically, if you're going to move the output files of Suite 2P, you want to move them all together. At least not the binary file, that's fine. Whatever you want to do with that if you don't want to keep it. But all these other MPY files, you need to keep them together so they load into the GUI properly. So I'm going to click on the stat MPY file, and it's going to hopefully open.
I don't see that one. That's 23 example data, so it would be 0.
You don't see it? So maybe people can't access it?
Hackathon/data.
Yeah, hackathon data, yeah, sorry. I think it's loading for me.
It's only accessible by your own guest account, the stuff in data.
And it'll take a second to load it. In this case, 10 seconds. Exactly. Well, maybe a little longer, but so Suite 2P makes the masks and everything for you. And it's also using, and all this stuff again, oh crap, I'm in full screen. I don't know how to get up here right now. And all of this stuff about the outputs is here in this, it's also in the wiki, but now it's also here, so you can see all the outputs of Suite 2P. So it's going to output an array of the fluorescence, which is what, for each cell, we click on a cell and we see its fluorescence down here. That's this trace, this blue trace. And then there's the fnu file and the spikes file. And fnu is the neuropil, and then the spikes are the deconvolved traces. And these are all shown down here. And if I had the right aspect ratio, it's not quite right, but you can kind of see these labels here. You should be able to see them normally. So they say blue is fluorescence, red is neuropil, and this grayish trace is the deconvolved trace. So these are the things that we show you in the GUI for each cell. And if we just, let's ignore the not cells for now. So there's two panels here that you can see. And on the left side are things that the classifier thought were somas. And they put them on the left side. And then on the right side are things that aren't cells. Can everyone hear me, or should I wear the mic? Are we good? It's fine? Okay. I should wear the mic? Yeah. People sitting right here are not supposed to answer. All right, so this left plane, so I have the labels up here too, it's a little confusing, but so the left panel are things that the classifier thought were cells. So we call these things ROIs. They thought that these are the ROIs that were cells. The ones on the right side are not cells, and then are what the classifier thought were not cells, but maybe you'll see some stuff there that, well, this zoom is very fast, that you might think are cells, and then you can actually right click on them, and they'll get flipped to the other side.
Wait, did you do registration metrics yet?
No, I haven't yet.
Alright.
I'm just showing them some things.
Hear it back here.
Huh?
We thought we just didn't hear it back here.
Very funny. All right. All right, so this is basically all of this information is inside the GUI, so you don't want to, so if you're going to move your files around, you want to keep basically all these files together, these 6 files that the GUI outputs. It's basically what I was trying to say. All right, now let's talk about registration, because Marius is very excited about it.
Okay, so I mean, the first thing whenever you just want to be really sure the registration works well, because it should work like, near perfectly, right? Because that's going to mean that all your issues afterwards are going to be small, right?
Hopefully.
I mean, it takes a weight off your, just to see that at least registration works nice.
And so in the binary view, if you have 2 channels, it will show the 2 channels. And there is an advantage to seeing the red channel because it's a structural channel in this case. It's pretty easy to see when things are moving on that channel. So I can play this movie, and even though like there is some lag from no machine, you can pretty clearly see things moving around. I can move, so I'm pressing play and pause with the space bar, so that just plays and pauses the movie. I can also use the left right arrow keys to move through if I want to. You can also click, so say I want to go to this frame here, I can just click my mouse here, and now I'm on this frame. And this is kind of where I want to be to check on things, because this is where I saw a lot of big. All right, let me tell you what this is. So these 2 shifts, these 2 lines are the shifts in X and Y. So I want to go to places maybe where these shifts are pretty big and see that the correction worked pretty well. So again, this is what I was showing earlier. So the left is the uncorrected and the right is the corrected. So let's zoom in on the corrected view and see that things look okay around here, and then all right, let's do the uncorrected view first. All right, let's see what happens. All right, you can clearly see things moving around. And there is some lag from the no machine, but you can see things moving. And then if we go, let's go over to here. Let's go to the same area with these red dots. Let's go back. Let's start over. Now, hopefully, most of what's happening is that cells are just becoming active or inactive. You can't really see large amounts of motion. And, I mean, you can try to trust your eyes with this, but you should also look at the registration metrics that we talked about to really quantify these things. Because, for instance, Z-drift, you're not really going to be able to see very well by eye.
How do you do that?
It's coming. It's coming. Okay. So one more thing. I don't think you can see this because we have 2 channels, but normally you can tell this trace here is a cell. So it's a selected ROI. Normally there's a circle on here that shows it, but I don't think it's showing up in 2-channel view. So, but normally if you just have a single channel, you'll see circles. You can select different ROIs and see their activity, and you'll see them on the field of view. But right now I'm just getting the activity, and I'm not getting to see where they are. Sorry about that. There's an issue to fix for me.
Is it like a maximal or minimal pixel shift or it just depends, I guess? I don't know.
There is a max pixel shift that we set with max reg shift, and it's set to 10% of the field of view. So we don't expect rigid shifts to be more than 10%. And then for max non-rigid shifts, we set it to 8 pixels. And that's something, I think it's set to 8 pixels by default. I'm not sure. It's 5 by default? Okay. So that's something you might want to make bigger or smaller. So if you're getting a lot of movement that seems like if your field of view is moving more, now that you've registered it, you probably want to decrease that. But if you haven't been able to correct enough, you might want to increase the amount you allow it to move.
Because I can again see from the metrics, right?
Yeah, so that's something you might be able to see from the metrics and also when you watch the movie too.
It should never move more after you register it.
Yeah.
If it moves more and means something else has failed, like, after looking at what's failed.
You might have to high pass filter the data or something too.
Can I ask though, so we use different zoom factors, sometimes 3, sometimes 4, sometimes 5. But then it would actually make sense, right, to then, depending on the zoom factor, change the maximum shift, for example, right? You actually would want to express it in microns, I guess.
Yeah, so you definitely could change it yourself.
Yeah, exactly.
I don't think, I think that's, yeah, that's a special, I wouldn't want to put it in microns, but.
So
I don't know, it's an option. We could consider doing that, but then the user would always have to tell us what their pixels per micron are.
I'm fine with computing backwards from microns to...
Right, but yeah, that is a good point.
So the 0.1, what is it? Like I have like max right sheets.
Yeah, so that's 0.1 times the size of your field of view. So like if you have 512 pixels, then you're getting, it's 51 pixels. That it's a max shift of 51 pixels. And if you're getting shifts bigger than 51, then it won't correct them, yeah. It won't correct more than that. And hopefully you're not getting shifts more than 50 pixels.
But
So
if they are bigger, then they'll be labeled as bad frame because they don't have a bad correlation. If any of the allowed positions, they'll have a bad correlation with the frame. And because of that bad low correlation, they'll be labeled as bad frame. And that was kind of
Can I ask just a question for my understanding? So, if you have shifts, right, shouldn't you kind of, your corrected image be smaller in the end than your 512 by 512? I
yeah,
haven't seen that so far when I run my own data. So is there something I'm not getting there, or right? Because naively, I think some pixels should fall off, right?
Yeah,
And
yeah.
for some frames you don't have data.
yeah.
So how is that handled, or how?
So with the interpolation with the non-rigid, that's taken care of, is that we do the interpolation, so everything will get filled in, but the edges will look funny. If things are moving non-rigidly, you'll see weird wavy things. And then with the rigid, we use MP roll, which literally just goes backwards. So you get the other side of the plane on the other side. It's just like, it's just what happens.
So that's also the reason why you don't.
You want to cross.
ROIs there on the edges?
Yeah, you don't want to take ROIs there, definitely, because, I mean, we could fill it in with anything, right? We could fill it in with zeros. It doesn't really matter. But yeah, you really don't want to take ROIs on places where shifts are often happening. So like in this case, shifts are pretty small. Like, I don't know why I don't have a scale bar here, but they're on the order of 10 or 5 to 10 pixels or less.
Right.
But anything more than that, I don't want to crop any less than that, because then I'll get crap, basically. And you'll be able to tell if you have ROIs that are just living on the edges. And there might be some even in this recording, but yeah.
So if you don't have any information about pixel size here, what is it using to automatically detect what it thinks is a cell?
So it actually, so what it does is, so when Marius talked about the correlation map and extracting these peaks, so it extracts the first, so it extracts a bunch of these peaks, and the first 100 that it extracts is what it uses as the average size. So that's what the classifier says, this is the average size, and then it says, it computes the normalized pixel number. So then every cell has this normalized value, and it prefers ones that are closer to 1, closer to the size of the top correlated things you find.
Sorry, that's done per session that you analyze?
Yeah, that's done per session. So that is one thing that goes into the classifier. Yeah, that's not that's dependent on the data. Yeah, 95% sure. I can double check though. Okay, so let's look at the registration metrics now. So if we go into the registration menu, there's another thing we can look at, which are the metrics. And now let's see. All right, so it looks like registration worked pretty well. So let me explain why I think that. So here's the first principal component. This is the difference image. So all right, again, we took the principal component across time of this pixel by pixel thing, and that's this line here, shown here. And then we take basically whenever this line, this principal component across time is big, we take averages of those time points. So this time, this time, this time, this time. These are all big. Take the top 100 time points, average those, and that's the top, that's the PC high, we call it. And then the bottom 100, that's PC low. So the bottom 100, like, frame at this time. And so then we take this is showing the difference, this is showing the merged, and then on the right, if you press the arrow key, you can play the two of them, and you can see the top and bottom flashing on and off. Oh yeah, I should call them top and bottom then.
Spacebar, right?
Yeah, sorry, spacebar.
Okay, and so this is where you want to use your eyes, right? Because your eyes will pick up motion between these two images if there's motion. and you want to find the kind of the right zoom level.
Yeah, sorry, I should say this too. In Sweet2P, everything to zoom is the scroll bar, and then if you get too messed up, you can just always double click and you'll go back to the center view. That's like the general shortcuts in all the windows you'll have. So I can zoom in a little more maybe to try to see a little better what's going on. I mean, and this mostly, all right, I'm zoomed in too much, but zoom is very difficult to control. Okay, with no machine. But yeah, for the most part, this looks like activity. This doesn't look too much like shifting, but I have to stare at it.
So you would look at the kind of medium bright cell.
Yeah, so if, especially like in this case, yeah, so it doesn't, you can try to look, yeah, at a cell that's just this
Right.
average intensity, yeah, exactly. That's still always on, and then it looks like it's not changing. And we quantify, oh, sorry. We quantify this. We take the top and bottom, so PC, the high PC, low PC images, the top or bottom, and we register them to each other. Or we'll actually register them to the reference image. And we say how far apart, or how different from the reference image is the top PC and how different from the reference image is the bottom. And then we see what this pixel shift is. And in this case, the pixel shift is 0.5. So on average, they're shifted 0.5 from the reference image.
That's the maximum. 0.5 is actually quite a lot, but it's actually only the maximum over the field of view.
There's only one number.
No, there's big numbers.
There's only one number, that's the shift.
So there's the mean shift across the field of view, but we're doing this with non-rigid.
Oh, okay, yeah, all right, so, oh, sorry, that's the non-rigid max, you're right, sorry, I have them wrong in my head, yeah, sorry. The rigid is this white line, which is at 0. Thank you, Marius, yeah, sorry. The non-rigid is at 0.05. So now we register to the reference image, and we see how much are the little blocks from the reference image. And that average is 0.05. And then, yeah, what Marius was saying, sorry, the blue line is the non-rigid max, which is the maximum possible shift of one of these blocks from the reference image. And you can see that this maximum shift is 0.5 pixels, which is pretty small.
Which is a lot, actually.
It's not that big.
It is a lot, but I mean, the reason why that happens if you zoom out is that some areas of the image are just pretty dark. So, you know,
But the
like
average is
arrow on
small.
the right, remember where we made this observation during Carson's talk that the arrows were kind of messed up and jumbled? It's because it's so dark over there, right? So, you know, the thing is, it doesn't actually matter if we register well there or not, because we're not going to have our lines there. It's too dark. Even if we do have it there, we don't lose it. So that will be where we'll get this relatively large pixel shift, because there's not enough SNR. But really, you know, the measure you want to optimize is the mean shift, which is around 0.05. And that's, you know, for this first principal component. So now what you want to do is go through the principal components and, you know, see if you find any one of them that is
And that's with the left, right arrow keys.
has a lot of motion, then that will be working. And you know, it's already summarized up there. So you can already tell, you know, all of them are pretty close to 0, you know, other than the maximum, which is always going to be because of that dark spot. But you know, you do this a couple of times, you go through your data, and you know, it'll look different for your data. When it's not working, it'll be pretty obvious that it's not working. The values will be larger, and you'll be able to see by eye, you know, that it's not just activity there. The other thing I was going to say is, you know, this view, you know, it tells you whether the registration works, but it already starts telling you, you know, how much activity you actually had in your recording, because these are the principal components of the activity. So if there's no motion, then it's all representing different patterns of cells that got active at different times. And so if you kind of nicely have, you know, a lot of cells kind of moving around between one end of the PC and the other one, and the different PCs are kind of different from each other, which they have to be, so it's pressing move through. So see, this is a different pattern.
The PCs are by definition orthogonal, so they'll all look,
Yeah.
they all should be different patterns of activity. Well, hopefully they're different patterns of activity and not different motion components.
Okay, so if this looks nice with lots of cells popping in and out, then it means that also the cell detection should be able to do a good job, because there's a lot of variance in the patterns of these observations.
it that the error with non-rigid is slightly higher there at max? With the first PC. Seems like after you did the non-rigid it meant higher.
You're saying the non-rigid shift is higher,
Yeah.
this red shift is higher for the first PC?
Yeah.
That they might be in that order in general, because that might be the way that they're actually going to extract the maximum variance PC might actually be the one with motion. Another option is that the max variance PC is actually when the mouse is running, which also might have motion, but it's a very small difference. So I wouldn't be too worried about it.
As long as these values are in that regime, even 0.2 maybe, like, don't have to worry about it. If they're larger, you can look into them. Even if they, if they're larger, but you look by eye and you can't really see motion, you're still fine. It's because it's a combination of like SNR as well, but just how much neural patterns there are as opposed to non-neural patterns. So okay, well that's it for registration stuff, right?
Yeah.
Okay, so I think the first thing to do for you guys on your data that you just uploaded and you're going to start running C2P on soon is just make sure registration works. So we should all go through this process with you in your data, and I guess we'll come around and take a look at your data sets. Tell you what we think. Did it work? Did it not work? Can we improve it somehow? But we want to be as confident as we can that this first step works well before we move on to the next one. And let's leave matters of classifiers and other cell detection stuff. Let's leave that for a little bit later. Let's do the registration first. Yes.
Sounds like a good idea.
The main thing I want to talk about is the classifier and how that works to label things as cells or not cells, or left versus right. Now, the first thing that you do is, you look at what comes out, right? And what happened here is C2P, I explained to you how it extracts all the ROIs, but without paying attention to if something is a cell or a dendrite or whatever. It doesn't know. It's just extracting things that have correlated pixels. And so there's another step after that that tries to look at properties of these ROIs to decide if they're cells or not. And that's what we call the classifier. This classifier right here is the default one for like all of C2P. It's a classifier we've built, based on some of our own set of data, works well on kind of the data sets we normally look at. It's actually not been optimized at this kind of zoom level for these cells. So it's not like the optimal data classifier for this data set. So what I'm going to do now for the example data set, and you guys can do it for if you've gotten to get cells on your data sets and you want to do it directly on that, that's fine. Or you could follow me on the example data set, or you could just pay close attention so that you can make sure you can reproduce this in 10 or 15 minutes from now. But basically, the first step is to train the classifier, is to have some pretty good labels for it. So what we'll do is I'm just going to go through and whatever I think is a cell, and for now I'm just going to move through here and get the obvious ones. Now some of them are not going to be so obvious, and you might have to switch back and forth between different views to get a good sense. Or you might want to click on it, and if you click on it, you'll see that. You know what? I'm going to close this. Oh, come on. There you go. Okay, so it has really good, nice activity. The blue trace, a lot bigger than the neuropil, the red trace. So this is going to be one of our best cells that was rejected by the classifier. So let's try to put it into our good set and see if we can train a classifier to do a better job. So you do this for a little bit. Sorry?
Why do you think it was rejected?
That big one? Right, so that's one of these. We'll talk about that later. That's something we're trying to kind of improve on. But cells that are not like your stereotypical kind of compact round ROI actually get
rejected right now by the classifier because they're, you know, they have dendrites and things like that, and so it gets confused whether it's a dendrite or a cell. But we can probably improve a little bit on that, and you know, with one of the teams, we're going to try to do that. Now, you know, this is like a really small kind of cell body, but I can recognize, you know, it gets harder, right? Like after you get, what I recommend doing is, well, you could do two things. You could like move through the ROIs one after the other by just pressing the right key, right? And that will just go through all of them. But that's probably not the most efficient way to do that. The most efficient way is, again, to use your visual system, you know, very high-tech technology that you have in your brain, and pick out a lot of things, you know, just kind of scan it, and you'll get a pretty good sense of, you know, what's a cell and what isn't. Yeah?
If you get an ROI that clearly looks like a cell, but if you look at the activity trace, then it's pretty much the same as neuropil. Do you intentionally ignore that because you don't want to include cells that have bad signal in your analysis?
Yeah.
Or do you include it because you want the ROI, the classifier, to find
right.
cells?
Yeah, that's a good question. So actually, that shouldn't happen too often because everything that makes it up to here usually has activity distinct from the background, from the surround. So it might be small activity. It might like be literally one transient over like an entire trace. And so yeah, I would take it in that case. It shouldn't happen too often at this stage. Okay, I'm not going to click through all of them. Just kind of give you a sense. You know, of course, some of these kind of dimmer cells that do have long processes, that actually makes them, we're more certain that they are cells because we can see the entire kind of anatomy of them.
So are you right-clicking to put them to the other side?
Yeah, I'm right-clicking. And as here at the top, you have these 3 buttons for the different views. You can either focus on the not cells, or on the cells, or on both. And if this isn't broken, when you click on both, they should both move together. So now it's broken for some reason. But you know, they would zoom in together, and so that would, I guess, help in some situations. But really, you'll probably just be in this not cells view, and you'll be scrolling maybe at this zoom level, and say, you know, things become, you know, that's the lowest in our cell. It'll take it. You know, you'll do quite a bit of this. All right, let's assume for now, you know, I haven't at all exhausted, you know, all the cells in there, and much less so discarded bad things here. But so maybe let's do a little bit of that too. Let's see if there's bad things on the left that we don't want. So I know this is maybe there's a cell there, but look at that. Oh, it's not very well defined. You know, this is probably too small. It's either okay. Well, you know, this is the kind where it will be pretty hard to know if it's a cell out of the plane of view. So I'm moving here between different views. I'm trying to find the maximum projection. There it is. So, you know, it's this thing here. Now, it looks too big to me to be an apical dendrite. So I believe there's a cell kind of sitting a little bit below the imaging plane. And maybe like this is the region of its apical dendrite. So maybe it's not cut quite well, I don't know. But in any case, if this went through with the analysis, it would not be the end of the world, right? But now that we've looked at it, you know, might as well reject it if you want to be conservative. Okay, so you know, you might go through, find other things. Oops. I don't know, what's that? Let's put that away, etc. And you know, you may want to do this carefully for like the first few data sets because you're going to use that as training data for the classifier. So you might, you know, want to spend a good amount of time. It will also, you know, probably if you just started the project, right, and these are the first data sets that you're taking with this particular type of recording, you'll be doing this anyway. So you're manually refining. Okay, so you got to a point where you're pretty happy about it. Okay, so now let me tell you also what happened. So the only thing that you've changed when you right-click on ROIs, move them left, right, is you changed its label from 0 to 1 in this file on disk that's called iscell.npy. It's literally just a a long vector of zeros and ones. It's actually two vectors. One is 0 and 1s. The other one is the probability given by the classifier to this. So that's the only, and every single time you press your right click, we're updating that file because it's just a very small file. So we just update it instantly. So you never have to actually like save the manual curation that you did. It's already kind of automatically saved.
Unless you want the MATLAB file. And then in that case, after you're done with manual curation, you can save the map file here. And it will automatically recreate this matcall.mat file for you with the right, like, with the iscell that you want.
Okay. Okay, so now we want to build a new classifier for this data set. Sorry?
Nothing.
Okay. Am I doing this right? Okay, we want to load an iscell MPY. And so we're going to load it from this particular data set we did right now. So we have one file. Now, if you're done, and ideally you don't just do one data set or one plane, you do a few of them, maybe like with some variability between them that would help make this classifier more robust to future data sets that you might have that might be a little bit different. And so, yeah, maybe you have multiple files like that you add to it. And then you say build, and it will ask you where to save that classifier. You should find a nice place to put it. I'll just put it here. New classifier, and save. Okay, so it was good. Okay, so now we can do two more things. We can either save this new classifier as a default, so then it will be the one that's used every time, the first time the data comes up, it will be already applied. Yeah, so let's just do that. Okay, it's going to overwrite our default classifier. Now you can always revert back to the default suite2p classifier, so you don't have to worry about that. Okay, let's overwrite it. And then you can also apply this particular one in the GUI. So we can click load in GUI, and it got loaded in the GUI. Now nothing actually changed behind here because it was loaded in the GUI but not applied yet. So to apply the classifier, well, we want to see the new labels, well, the new probabilities. So I clicked on this probabilities bar here, and you see it colored every cell according to its probability. Now, most of them have gotten probably pretty similar probabilities to before, but there'll be some changes. And so now if we apply the classifier, what will happen, right, everything above whatever number we set here, everything that's maybe green to red will move to the left. Everything that's cyan to blue is going to move to the right. So that's all that will happen. So I press enter just now in this box where it said 0.5. And so it'll take a little bit of time to redraw all the cells in the different places. And looks like it's updated now. Okay, so yeah, so there's threshold is 0.5. Everything was moved. Now, if I had done probably a better job, we could have of labeling this manually. We would have gotten a sense of that, the fact that whether it improved or not, but at this stage we probably can't. But that's basically the workflow. So that's what I wanted to show you. And there are still, so here, these guys, which are some of our best cells, they were still misclassified. So that again has to do with these like little processes coming out of them. So hopefully during this workshop we'll improve the classification of those. But that's kind of a known issue. Like right now, it's looking at things according to kind of how compact they are and how similar they are to some somas. All right, that's the basic logic. So you guys can play with this, either on the example data or your own data. Any questions you have right now? Yes?
Does any of those metrics help you determine whether it's a cell or not?
Yeah, good question. So these are, most of these metrics are the ones we actually use in the classification. And so I think by default, we use, I want to say skewness and like footprint. And I think it's these three. They're all a little bit redundant with each other. the size, the footprint, and the number of pixels are a bit redundant. They all get kind of normalized to the field of view, so it's not just the number of pixels, but it's normalized to like the average number of pixels for an ROI. That's what goes into the classifier. But yeah, it's built based on these numbers. And what we're thinking of adding during this workshop is just kind of build another classifier that's based on the shapes of the cells. And things like that should be a lot more easily identified by shape than by these particular metrics.
So when you showed us the presentation in the morning, it seemed like most of the cells were around the skew of within 4, but then I saw a few cells when you went through that the skew was 12 and 14 and stuff like that.
Yeah.
Does that mean anything?
I think if it's super high, it's usually like maybe like this one. Let's see. What this is like 6. So it's because it had, skewness is this thing that measures how often do you get outliers. So the most skewed trace would be one that's perfectly flat and is just on one time point. It's large. That would give like skewness 100 or something. I don't know. So basically, the higher it is, the more it means that you have relatively few of these big amplitude events, which is the kind of statistics you look for in your cells. Not all the time, right? Sometimes you get high-firing cells that have a lot of signal, and those might actually be the ones responding to your task or your behavior or something. And so those, maybe a little bit more like these ones here, right? So they clearly have transients. They're just not as sparse as the other ones. And so maybe the skewness now, it's a little bit lower. It's 3 now. Now, if you get into the skewnesses of like 0.5 or something. Actually, let's switch to skewness, and we're now coloring the cells according to skewness. And so, okay, we can take the top cells. So if you see here at the top, it from whatever color, however you color the cells right now, if I press this button, it will select the top 40 from that particular map or sorting. So if you look at these top 40, right, these are, basically the cells that just had like one transient, so those are the most skewed. And then if we take the bottom 40, now let's see. Right, so these are going to be the ones that look least like they have transients. Now, for these cells that are relatively not skewed, it could be one of two things, right? Either they have a lot of activity, but it's kind of going on all the time, or they don't really, and they represent mostly kind of like the neuropil. Already you can, I don't know what's happening here. Look, these two are going against each other a little bit. But a lot of them, they have quite a bit of correlation. So I think what we're seeing here is that, one set of cells are pretty close to the neuropil, and we're seeing them as similar. And then every now and then there's one of these ones that are not, they actually have good signals. They're just ongoingly high activity.
When you're in this view, can you see the neuropil trace so that you could kind of look?
Not together at the same time, but we could switch to it. So if I switch the neuropil trace, right, so that's a lot more homogeneous between cells. So yeah, just switching back and forth between those two, it doesn't look like.
That's a neuropil chart. So you F versus F.
Yeah, F and corrected are a little bit different, but I think it's. I mean, do any of these traces look like the neuropil? I don't know, maybe. You can probably look more closely. I mean, you can just select a few of the cells one by one and go through them. And for this cell, for example, it's actually nothing like the neuropil. Right. So this is one of the examples that it has like transients riding on top of transients, riding on top of transients. It's just activity throughout, right? And it's kind of nicely modulated all over the place, all the times. And it's not because of the neuropil. It's just this cell had a high firing rate. So its skewness is 0.25, but it's not a bad cell. It's a good cell. All right, any more questions about this view? So yeah, I mean, with the GUI, I don't know if we really gave you a GUI tutorial, but yeah, these colors, just briefly, the colors, you can switch between random, skewness, compactness. So these, skew, compact, footprint are the same things printed down here. And then you can color the whole field of view with them. Footprint. Footprint is pretty useful. So usually you don't want things that are either too large or too small. And so if you look at the two views, you'll see, and probably the classifier in this case was very highly biased by this footprint because it mostly got us these cells with footprint 2. Again, it's probably an indication of the classifier being not quite well adjusted to this field of view, whereas we get all sorts of different sizes on the right. So the footprint is just kind of the, how many pixels around our ROIs are correlated with it, whether they're in the ROI or not. How big of an area does this ROI correlate with? If it's a really big area, that's probably not good because it's probably like a neuropil. If it's really small area, then it's probably a process like a dendrite or something. And then, okay, channel 2 probability. Let's not talk about that. Okay. Who knows with it? Okay, classifier. We already looked at that. Okay, now this is where Sweet2P starts to become more like an analysis tool. And of course, it's not meant to replace like actual real work that you guys should do outside of the GUI, like real analyses. But it's meant to like start giving you a first sense of what the data is doing. It might help you to, it might help you to design the next experiment for the next day, right? But it's not going to go in a figure in your paper or anything like that. So here I clicked on a cell, and what's plotted now in this correlation view is every color is the correlation of this cell with the cells around it. And because I happened to be in the neuropil activity mode down here, so now everything is computed based on the neuropil, not on the cell's activity. So that's why we're seeing this kind of pattern where we actually over a big area we can see the neuropil is highly correlated, right? I mean 0.99, right? Like above. Like even yellow is a 0.8 correlation. Green on the edges here is a 0.5-ish correlation. So that's roughly the spatial decay of the correlation of the neuropil. Click on a different cell. This one's a little different because it's a lot more locally correlated, perhaps. Click on a different one. Let's see. Okay, and so on. You can get a sense of the spatial structure of the neuropil in this case. Now let's switch to a different activity mode. So we'll just take the neuropil corrected traces. So F is the trace of the cell. That's the blue trace in the bottom view. Red is the neuropil, so F new. And then the neuropil corrected is the third one. So that's the default that you get started up when you load Sweet2P. We're going to go back to it. And now the correlations have updated, and you can see there's no more spatial structure like at all. There's a high correlation here with the cell that's basically overlapping. So that we can probably be pretty sure that it's well, we can be super sure, but it's probably due to the overlap that it's such high correlations. And we can click on other cells, and we can play this game to try to ask, can we find any cells that are correlated with their neighbors or not? This is doing visual stimulation, and this is in V1. So cells are retinotopically matched. So, it's, this is natural images, I think. Okay, it's natural images. Anyway, so basically my point is that, most of what the cells are responding to, whether it's... Are you sure?
Yeah, this is the high motion example.
Okay, never mind then. It wouldn't look too different. Okay, I have an example with visual responses in just one moment. But okay, this is spontaneous activity, so yeah, there's basically no structure in the patterns of correlations between cells. Now, you can do other things like start grouping cells together. So you say, if I have a bunch of cells that are highly correlated, why don't I just start to select them, kind of one after the other. Let's see.
Or you can select the top 10.
but it's not, it doesn't, it looks like the shortcut isn't going through. So if you keep Control pressed, then maybe you'll have better luck than me. You should be able to select multiple regions of interest. But
Shift also works now.
what does?
I implemented Shift as well. Shift click also works now.
The shift click also doesn't work here. I don't know if it's because it's to no machine and keyboard shortcuts are not forwarded or something.
Why don't you do the top 10?
Okay, right. So we're in a color map. So we could say, let's take the top 10 cells most correlated to this cell. And so I'm selecting this button up here. Did that work?
Press enter. If you change the number, don't press enter.
All right. So here's the top 10 most correlated to that cell. And if you look at their traces, then yes, I can see that they're all active. At this time, maybe also a little bit here. Maybe there's some coactivity here. So that's why all these cells are active. And this is not a very long recording, right? So it could be by chance that these cells happen to be active during that period. Although it's a little, I'd be surprised, right? I mean, they all have actually these nice kind of bimodal responses. All right, already looks pretty convincing. Maybe they're responding to similar kinds of things. Now, the other thing that happened when I selected 10 of them is the color view now updated to, so each cell is colored by the correlation with the average of these 10 cells. So if we actually repeat this process now and say, select the top 10 of this in this view, it will kind of keep iterating basically this way to give you, to kind of, essentially you're doing like a form of kind of manual clustering at that stage, right? You're kind of extracting groups of neurons that are correlated to each other. I guess I would have to press 10 again. Okay. Anyway, so that's one of the things you can do in the GUI. The other visualization that we have, whoa. Okay, I'm lost. I double-clicked. I'm back. So if you select a single cell, and then you go to visualizations, there's this algorithm that we call Raster Map, because it makes a raster plot, well, a raster image, and because it's an image, I guess we call it a map. It resorts the neurons along the y-axis of this plot according to the pattern of their activities. So we have our matrix of neurons by time points, and this matrix is perhaps too big for us to look by eye because we just have too many neurons or too many time points. And so what we're going to do is we're going to change the order of these neurons along this axis so that neurons with similar firing patterns are put next to each other. And you can imagine what will happen then is we'll start getting kind of a nicely progressive patterns of activity where maybe it will start in one end with one pattern and we'll kind of gradually morph into a different pattern and a different pattern and so on, giving us more of a sense of what are the biggest patterns of activity in the data. So I pressed that button up at the top left, and it ran it. And so this is now, it's the exact same data. All I did is I changed the order of the neurons along the y-axis. That's all I did. And I did a little bit of smoothing in the y-axis too, which is, I guess, not so important for 1,000 neurons, but maybe more so for like 10,000. OK, and so now what we have here is groups of neurons that do similar things. And if we actually knew what our data set was about, then we might be able to say, or to think, well, I started my visual stimuli is somewhere, so maybe like around here, with just giving it as a thought experiment, right? So if we had started some imaging, we'd see at some time point a different set of patterns would emerge because of the stimuli, right? Or because of the behaviors or whatever the mice are doing. And so it would kind of give us a sense of which neurons activated in response to what epochs of the experiment. Okay, and that's, we call this a visualization tool. You can also, you see it gives you multiple groups of neurons along the y-axis. It's not just, two or one group of neurons. You could also change it to sort the neurons by the principal component. And so now we're visualizing PC1 of the data. And so what you can kind of already see, well, this is, it's a single principal component. So, the neurons are sorted from top to bottom according to whether they like to fire at certain times or at the opposite times. There's actually kind of a push-pull kind of thing going on. And because we know, I guess because we think this is spontaneous activity, we know these are just kind of these spontaneous activity patterns that are happening in the mouse visual cortex, just have to do with the mouse's kind of own behavior and its behavior on the ball. So we kind of, we know by now what these patterns mean, but then you'll run it on your own data set and you'll see some patterns emerge. You'll want to see what parts of your experiment can you correlate those to. And this view is going to be a little bit different whether you run it with spike deconvolved data or with just neuropil subtracted fluorescence. And we haven't talked about deconvolution because you guys had enough on your minds already. But I'm just going to briefly introduce it now, and again, we can talk a lot more maybe tomorrow about it. The deconvolved trace is what you see at the bottom here as this kind of much sharper trace that's mostly 0 most of the time, and then it's some positive number otherwise. And that's an algorithm we run on the kind of the neuropil subtracted trace, so not quite the blue. So the blue minus the red, that's our estimate of the fluorescence from this cell. But it still has a problem, right? That it has this kind of long time scale of the GCamp that we know about that we'd like to get rid of, because we'd like to localize our events in our fluorescence kind of in time in a more well-defined way. And so that's the process that we call spike deconvolution, or you might have heard of it as spike detection or spike inference or things of that sort. But at the end of the day, it's trying to be an estimate of the spike rates of the neurons in the different bins here. Now, it's always going to be a noisy estimate because the data only says that much about the underlying spiking. There's always noise accumulated along the way from the microscope, from the algorithms, from all the processing going on. So, this is kind of our version of the best version we found, at least, to get closer to an estimate of the spikes. The main reason we like to use this is it localizes patterns in time. And so in particular, if you're doing some kind of, if you're doing things where you show stimuli, let's say, at 1 hertz, one after the other, and you're using GCamp, let's say the slow version, then the GCamp will not have decayed completely by the time the new stimulus is presented. And so it'll kind of always ride on top of the previous stimulus. And so if you can at least deconvolve these patterns in time, you'll get enough temporal localization that you don't have to wait for the GCamp to fully decay. You can kind of start trusting the temporal localization of the deconvolution algorithm. And of course, if you have like a behavior, that's even more important because behaviors are very fast in mice and all animals, I guess. And so there you have more and more of a temporal problem, right? You want to localize things in time as much as you can and kind of undo a little bit, this problem of long GCamp timescales. Any questions about spike deconvolution in general? I mean, we'll go through the details of exactly how it works, but I just told you what it's trying to do, basically. And it can,
I have a question.
Yeah.
My understanding from the Chen 2013 paper was that when you have, like here, if you have like a super big calcium transient, that's not, it sounds like that would count as like one spike, but it's probably a burst.
Mm-hmm.
And I'm just confused by
Yeah.
how you are supposed to think about like a giant transient versus like huge transients.
Yeah, so there will be always an amplitude of proportionality here, right? So this is what we use. We don't use zeros and ones because for 2 reasons. One is what you just said, right? In one bin, we know that some of these huge transients are usually because of multiple bursts, multiple spikes in it. Now, we can't tell you exactly how many spikes there are. We lack a few things to be able to do that. We don't even really have a calibration bar to tell us, one spike should correspond to this much calcium. that's actually really hard to get from single cells, to know exactly, even on average, how much df over f should correspond to one spike. So, we get a big bar like this, and we can tell you that there's 20 spikes here and 1 spike here. But what we can tell you is that there's probably roughly, 10 times more spiking here than here. So the magnitude matters, right? And roughly, to the extent that GCamp is linear, which it's not, but it's a first approximation, the magnitude should scale with the number of spikes. And that's the kind of the way you want to use it. Basically, it's always, think of it as if you had taken your nice electrophysiology data, it's a sequence of 0, and then some bins are going to have 1 spike, a few are going to have 2. Very rarely, maybe you'll get a burst, you'll get 10 spikes in a bin. And so you have your nice bin spike data, and now you add noise to it, like a lot of noise to it. That's roughly the statistical structure of this trace. Yeah.
Oh, my impression of the convolution was like old days. There's a paper, Nature method, Rainer Friedrich used like a in vivo patch time recording of each cell, and they determine the decay constant. In this case, he also measured calcium activity, but it's just the same calcium indicator, but different cells can still have very different ways of handling calcium. I wonder, I mentioned earlier today that in my case, some of my cells have extremely long decay. I wonder if it's a specific way of how they handle calcium.
Yeah, so it changes cell type to cell type, right?
In this case, it's like 1 tau for all cells.
We set it right now in C2P. There's only one parameter really that you're setting, which is the decay time scale, and it will be, by default, it will be the same for all of them. Yes. Now, if you had some, perhaps, yeah. So we've also we have one of those papers that I put up there at the beginning, is where we try to see how bad does the deconvolution get if you're wrong about your time scale by a factor of 2 maybe, or 3. And that's actually not, it barely decays the performance. So as long as you're kind of, maybe within a factor of 2 or 3 of the true time scale of your indicator, the answers shouldn't change too much. It's pretty robust to that. That said, if you do have some a priori reason to think, oh, this cell class should have a different time scale, so I'm going to modify, the parameter in C2P, and I'm just going to use that time scale. I think that will be absolutely fine. Yeah.
So some cell types, they have a permissive internal store of calcium in response to calcium intake. It would also show as a huge spike here.
That's a good question. I think that the whole process, it's a lot smaller than the influx of calcium you get from a spike. Unless you're thinking, I don't know, maybe there's some cell types that are very different from that, but that's my understanding. Most of these really big transients will correspond to spikes. Some of the small transients might be like what you're saying. Yeah.
How much do decompose data would change if I use different calcium sensors?
You would just, so there's one parameter you have to set yourself, which is the time scale. And so, we've tried it on fast and slow and medium, and it's, if you set the time scale right, you get, and by right, again, I mean within the order of magnitude, right? You can't be too wrong, but you get better answers. You get a better estimate of the ground truth spiking. Well, first of all, if you do deconvolution at all, and if you do it with roughly the right time scale. Where do we have the ground truth spiking from? Well, we have it from a few cells that were patched here and there. A lot of them here at Genelia in the Genie program when they were making the sensors. Some of them by other people elsewhere. So together they've put these data sets part of the Spike Finder challenge, if you guys have heard about that. Okay, so yeah.
What kind of quality metrics do you use to evaluate how good the deconvolution works?
That's an interesting question. So I don't think, other than having actual ground truth, right, which would be a clear objective metric that these algorithms are evaluated against. And what we actually did in that paper, let's see, I don't know if Carson has my slides here. No. Okay, well, so it's a lot harder without ground truth to know if you're doing well, right? So one actual metric that we found was useful was to ask how well, so what was the signal to noise ratio of the cells in response to stimuli? So the problem, right, so we show a lot of stimuli, they're coming one after the other, and if your kind of transient is always riding on top of some background activity from before, and your stimuli are coming kind of in random order, right, then you're always going to have this extra noise added to your stimulus response. And so if you do your visual stimulus SNR metric, like how peaky are your tuning curves, or how reproducible are your responses between 2 repetitions, so those are measures of SNR, because you've added this extra noise, they'll be lower than if you deconvolve and localize the entire part of the calcium that corresponding to a single stimulus should come just to that bin and not to the nearby bins. So then you kind of remove that extra noise. So we've actually, in that paper that I told you where we just look at spike deconvolution, we found that there was actually a very good metric, kind of a stand-in metric when you don't have ground truth. That's actually kind of a good second best thing you might have to evaluate them. Okay. Go ahead.
So I have the select top and select bottom in gray. How do you activate those buttons?
Yeah, sorry, so you have to be only in cells here.
Oh,
I guess you.
So in both view, you don't decide you want, so that's why they're only.
Another
Yeah.
question. I'm not able to use right click to send the cells to either side.
It doesn't. You right click and they don't go. Okay, we'll look into that.
You use 2 fingers. You use 2 fingers.
I did that, so I changed my mouse settings a couple of times and tried different things.
Okay. That may have to do with forwarding through the no machine remote interface. Maybe that's the problem. We'll look at that. Okay, one more thing I wanted to go through before we end this was what are I don't think we went through all the parameters. So you guys wanted to know what every parameter does?
Yes.
Okay, probably even better. So the first thing I can tell you is if you mouse over it, you'll get a little description of what the parameter is.
So in general, like 0 is inactivated and 1 is yes.
Yeah. Mm-hmm.
Yeah, for Boolean variables, that's the thing for true form.
Okay, so just going through them here. Number of planes, okay, so some of them are self-explanatory. So number of planes, number of channels. So this is where your functional channel is in a multi-channel recording. This is the channel where you extract your fluorescence from. So the traces that you see at the bottom here, these blue traces at the bottom, those are always selected from the functional channel, which is defined here. And it might be your first channel or your second channel, so that's where you change it.
This one is not 0 phase. This is 1 and 2. Functional channel and also aligned by channel.
Yeah, I mean, yeah. 1 based. It says right there, it's 1 based. Okay, so 1 based as opposed to like 0 based, which would be like the Python indexing. And then tau is the time scale. There's also these descriptions of these parameters are going to be also on the Suite2P website. So if you go in, it was right there?
Yeah, I ported it. Go back. No, 1, 2 back. Yeah. Click on the settings on the left. I put it in there, but it might not.
Okay, so they're mostly described here. But also I think, okay, you moved them completely here? They're only here now?
Yeah.
Okay.
They're also on the website, but they're the exact same. I copied and pasted.
Okay, so they're also on the GitHub. If we go on the GitHub and scroll down, okay, so this is actually how you would format these parameters if you wanted to run C2P outside of the GUI, and then you'd have access to like all of them. Right now in the GUI, the important ones have a corresponding one in the GUI, but some of them don't. And I think in some of the cases, you might want to change one of these unexposed parameters, and so we'll go with you guys over that. Okay, so this is where all of them are, but then what you just saw, I guess, Karsten is shifting the documentation from the wiki, which is where it used to live. And so if you go to the wiki, to this like, website basically that's built based on the code in an automatic way. And so if you look at input format, no, settings, ops and py. Okay, so most of so especially for the Python version, like most of the documentation and most knowledge about the Python version is in the wiki. We are going to write a paper hopefully pretty soon about it, but until then, everything is centering around the GitHub repository. And so all the parameters are here. I will still go through them one by one. So tau, the time scale of the GCamp sensor, well, whatever sensor you're using, it's the time scale of decay is just one time scale. We found that using 2 time scales didn't really help, so it's easier to just set 1 time scale for the decay of the GCamp. Again, whatever sensor you're using, not necessarily
That only to the deconvolution step.
It's actually used in one other place, which is where we actually, to make the cell detection run faster, we bin the movie according to the time scale. Because if consecutive frames are basically correlated because of this long time scale of GCamp, they're not really independent new samples of the data. And so binning them, we're not really going to use a lot of the signal to noise ratio to do the cell detection, and it helps us a lot in the speed of that. So we actually bin at exactly 1 tau of the data. So if our data is recorded at a sampling rate fs of 10 hertz and tau is 1, then the movie that actually we do cell detection on is binned at 10 frames.
And so why would you change that? So what should happen in the outputs that you would think, let me change my tau?
Well, I think if you're in a well-defined brain area where you roughly know how things are, there should barely be a reason to change tau other than based on the GCamp sensor, right? So fast, medium, slow. Now, even in Cortex, if you're recording interneurons, different interneurons have different kind of calcium buffering dynamics, then you probably already know a lot more than me about that if you're studying those cell types. So you probably know if you should want to make the time scale longer or shorter based on that. If you're studying interneurons that have these high firing rates, not very clearly defined transients, you might want to skip the convolution altogether because it's even more worrying there. Yeah, so roughly, for GCamp 6 slow, I don't know, 1, 1.5, 2, they're probably all pretty similar. If I had to give an average, GCamp 6 slow, maybe 1.3 seconds, GCamp, and the corresponding 7 ones too. The medium would be like 1 second. The fast would be like half a second or something like that. So that's kind of roughly like when the Chen paper, that's what they measured as the average. And again, it changes a little bit depending on whether you have more spikes in the bursts or fewer. So that's kind of a rough average of the decay time scale you expect for an average sized burst. All right, delete bin. So this is if you want to delete your binary file that's created by C2P. So for example, if you don't want to fill up your hard drive, you just want the results and you're doing batch processing, you might want to change this to 1. Do by the phase. Say again?
Why would you want to keep the binary files?
Oh, sometimes you just want to look at them like we just did, right? Just make sure everything ran correctly. If you want the algorithm to try to estimate the bidirectional phase offset, so oftentimes the 2P line scanner goes one way and then back, and then you have to figure out the phase offsets between back and forth. The microscope or whatever program you're using usually does this for you, but sometimes it doesn't. It can be small offsets, so it tries to estimate that. Or you can set it by hand yourself if you already know what it's supposed to be. So that's the last parameter there. Now, On the output side, whether you want the algorithm to pre-classify the ROIs and just throw away everything that's not classified as a SOMA. So, because you've reached the point where you're not even going to bother with those, you're just going to take the automated output of the algorithm. So you can set that to 1.
0.5. It's a probability.
It's a probability, okay.
Set that to 0.5, for instance, or 0.3, or whatever. Don't set it to 0.1.
Okay. Set it to 1, you won't get anything left. So that's the threshold on the classifier, the probability threshold. So it works the exact same way as the threshold that we looked at in the GUI. It's just that it gets applied right away. So it makes things faster because you don't have to extract fluorescence from every single ROI. You don't have to deconvolve every single one, and so on. So it's a nice way to like speed it up further if you're already confident that you like the classifier's performance. Okay, combined is whether you wanted to create a combined view where if you have multiple planes or if you have like a mesoscope bigger field of view that you want to tile together, you can do that. Actually, I'll show you an example of loading a mesoscope field of view in the GUI. But that's just for display purposes. If you want to create the registration movie as a TIFF files, you can set this to 1. But really, you get pretty good. Just the movie player in the GUI is already pretty good for what you normally use the TIFFs for, just to see by eye if there's motion. Same for channel 2. If you happen to have different pixel resolutions in X and Y, which we happen to do, then you can change the aspect ratio here. If you want to do registration, which channel you want to align to, so again, 1 or 2, you might be wanting to align by a non-functional channel, right? So that will be the red channel, in which case, maybe this will be 2 and the functional channel will be 1, or vice versa. How many frames to use for initialization for computing the target frame for registration. So there's the trade-off here is when you want a nice crisp target because you're going to align every single frame in your movie to it. So the better job you can do on this target initially, the better your registration is going to be throughout. And so a set of a few hundred frames is already pretty good in most cases. If your data is relatively low SNR, you might want to use more. Just to take advantage of averaging over frames, right? To remove some of that shot noise. But yeah, with more frames, it will get slower and slower because it's doing this iterative optimization, going like 5 or 10 times and realigning every frame to every other frame, to the target. The batch size is just a matter of performance, I guess. So as you're doing registration on the entire movie, maybe you have 100,000 frames in your movie. So it can take a while. So you want to increase this batch size up to the point where you run out of memory, basically. So 500 is maybe a good, maybe 500, 1,000. It probably doesn't make sense to increase it much more than that. But that's how many frames at a time it's going to register.
Would 1,000 frames be faster than 500 if your memory can handle it?
There'll be scaling up to some point after which it will plateau. I think it plateaus around 1,000 maybe or so. So now there's a parameter here for smoothing. Actually, maybe you should try that. So especially when you have lower SNR, it can help to smooth the image a little bit. So there's some default parameter for that, and you can try increasing that for better SNR. This is where we set the limit on the registration shifts. So kind of if you set the limit to 0.1, that the image can't move more than 10%. And if it tries to, it will just go to some random place. It will basically be classified as a bad frame. Yeah. Yeah, I moused over it. It says right there. Okay, this is a threshold on the bed frames. So for this threshold, 1 doesn't mean the threshold is 1. It has some, hard-coded value in there. I don't remember what it is. But you can adjust it relative to this value, right? So if you want more of your frames to be called bad, because they're not well correlated with the reference, then you increase this threshold. If you want to be more permissive, you decrease the threshold. Whether you want to keep the raw movie, so you already did this to get both the raw movie in the viewer and the aligned movie. Non-rigid is on by default. And by default, it comes with block sizes of this big in pixels, 128 by 128. Now, that's the only reason why that's the default size is that a lot of people's recordings tend to be in the order of a few hundred pixels by a few hundred pixels. So that's roughly a good size. But if yours are very different fields of view, then you can consider changing these block sizes to roughly maybe, this is the spatial scale at which you expect motion to be, roughly similar across this big of a field of view. So within 128 by 128 of my, let's say roughly microns in this case, I expect motion to be roughly the same. And then I allow it to change more and more the more I go away from this block. Okay, there's some more low-level parameters that we can talk about. This one, max ref shift, non-rigid, and R stands for non-rigid, is the equivalent for the other one, for the max ref shift, but this is for non-rigid shifts, and it's 5 pixels by default in every single block. But that's after we've already done the kind of the big rigid alignment. So the rigid, most of the movement you'll get in all your movies are going to be rigid, maybe tens of pixels, let's say, and then there's going to be a little bit of non-rigid left after that, which we correct essentially with a second pass of non-rigid alignment.
Sorry, what were the units for that? What does the 5 actually correspond to?
Pixels. Yeah. Okay, there are some 1P specific settings here that I'm not going to go into. Basically, mostly for like things like miniscope data and other kinds of 1P, I guess. It's just generally things are more blurry, so you have to be. Remember, for registration, we rely mostly on high frequency features. So if things are blurry, then you have to kind of worry a bit of how are you going to accentuate those high frequency features to try to make it easier to register the data. So most of those settings have to do with that. And then we move to ROI detection. Whether you want to do it or not, again, 0 or 1. So I think by default this is 0, but we want to run the sparse mode branch. So we switch to 1. Sorry, not branch, the sparse mode algorithm. We switch to 1. Hopefully in the near future this will become the only option. And then this other diameter parameter won't matter because actually the sparse mode infers automatically the spatial scale, which is why the spatial scale parameter is set to 0. If you want to infer the spatial scale by yourself, just force it because you're not happy with what the algorithm determined, you can change it here. Connected is kind of like a post-processing step. After you've extracted all the ROIs, if you want to like clean up kind of random pixels maybe that have been put into the ROI for no reason, you can just have a connect. You can set this to 1, and then we'll only extract the connected region of this ROI. So all the pixels have to be connected to each other through a path. This is if you want to extract more or fewer ROIs. You can adjust this threshold up or down. So if you set the threshold higher, then remember we're extracting ROIs starting with the biggest ones, the brightest ones, and we gradually, and we peel them off. And as we kind of get into like the kind of more noise, then we're starting to extract ROIs that are smaller. At some point they'll be below threshold, and that's where we stop. And so if you want this threshold to be higher, you can just adjust it here, by a factor of 2, or maybe you want it lower because you want to get more ROIs. There's some more options, again, to do with this kind of post-processing where if cells are like literally on top of each other, then actually we discard every cell that has a lot of, more than 75% of its pixels are overlapped with another ROI. We just throw it out. We just don't even bother. Or you can change this if you wish. Okay, number of iterations. So this has to do with this kind of iterative process I was telling you about, where we extract cells one after another. I think by default, maybe it stops at like a pretty large number, like 4,000 or 5,000. So usually you shouldn't have to change that. And finally, this is the high-pass filtering option. This is what we use to get rid of the neuropil in our cell detection step. So this is literally in pixels, the high-pass filtering value. Again, it should scale a little bit with your zoom level, but this one is? Yeah. Okay. This one's high-passing in time. I guess in space, there's also high-passing being done in space, but I'm not sure which parameter controls that. I think the inferred diameter is going to control that. Okay, and again, this high-pass filtering in time, essentially because we want to emphasize, well, I don't think it will matter so much for, this is a very large number. I don't think it's in seconds, and I don't think it will matter much for like 2p data. So don't have to worry too much about it. It's more for like data where the background is really high and you kind of really want to high pass kind of the slow fluctuations out to find actual cells. Okay, let's see, what else do we have here? There's some options for if you want to allow overlap between pixels or not. By default, it's 0, so this is just that we throw out all of those pixels of ROIs that overlap each other because we don't want to bother with the problem of demixing them, of telling apart which cell that signal is coming from, which is usually fine, right, if your cells are sparse enough that you only have small overlaps. But if you have a lot of overlaps, you might want to think of other ways to do real demixing. Again, the inner neuropil radius and pixels, these are just kind of, rough settings to allow us to pick, at least, I don't know, 350. If we can find at least 350 pixels around every cell that are not occupied by another ROI, then we can have a pretty good estimate already of the neuropil. And it almost, it doesn't really matter which pixels, because in most sparse enough recordings, there'll be a lot of these pixels. And again, special case, when things are dense, it might become really hard to pick out pixels for the background. So let's worry about that separately with team dense. All right, and finally the deconvolution. So the neuropil coefficient here that's being used in the convolution, by default it's 0.7. So that's the first thing that happens. We do a neuropil subtraction. And after we do that, we do a baseline correction in addition to that. So basically to estimate, let's look at a trace. We want to estimate the time varying baseline of these traces so that, only the spikes, the transients stand out. Usually if you subtract out the neuropil and you don't have other sources of slow motion, you'll be fine, but you might have some bleaching, you might have some movement, maybe even a little bit of Z movement will result in what looks like a baseline shift. And so that's what this extra step here is for. Now, just to be clear, the traces that come out of Sweet2P, the F and F new, those are not processed in this way. This is just what happens to get the spike deconvolved traces. And so, when you guys, let's say you want to analyze the fluorescence traces from Sweet2P, you should still worry, for yourself, how should I baseline these traces? Do I need to do some temporal baselining? Is a single F0 maybe across the whole thing enough? Do I want to do DF over F, or should I just do DF? These are settings that we kind of left for you because depending on what you're doing with the data afterwards, you'll want to use a slightly different processed fluorescence. All right, and that's it. And as soon as so both of these are just for parameters for windowing that baseline. And then the deconvolution runs through. And these are it. That's it. That's the exposed settings. There's a few more that are hidden, but they should have even less of an impact than these. All right, questions? Yeah.
When you export, when the P spits out the F and PY, that was not corrected.
No.
So if you're going to subsequently use that data, you have to include that in your analysis.
Yes. So it splits both an F and an F new NPY.
Yeah.
So there's two matrices. They're exactly the same size. And a third one, that's the spikes.npy, also the same size. So all these three are just as big, neurons by time. Okay. Okay, while we're still on the same page, we all did registration to begin with. And so just wanted to, I think in one case we found an example where there was a lot of Z movement, I think in Sarah's data. And so, I think it would be good right now if we'd go over kind of ways to try to remove Z movement, basically. Because, if you get to the stage that you have the data and there's been Z movement in your data, there's basically nothing you can do, right? You've lost, if your plane has moved from where the cells are, there's no algorithm is going to correct for that and give you back missing data. So this is like the one step that you want to be sure that you've done everything you can already in hardware, in the actual setup that you have to reduce it as much as possible. So I thought we'd just, have a little, I think this is something that, because you guys all do imaging, this is something that you've already probably dealt with. So just kind of collecting everyone's tips and tricks for how to keep recording stable, I think that would actually be pretty useful.